Welcome to the Formation of Connections tutorial. Neuron morphology is the shape, structure, and physical characteristics of neurons. For example, the size and shape of the soma or body, the number and arrangement of dendrites, the length and path of the axon, specialized neuron structures such as axon terminals, synaptic connections, or myelin sheaths. Neuron morphology can be studied from two perspectives. First, we have the cellular structure, which is the study of neuron morphology, including detailed examinations of the cell body or soma, which contains the nucleus and is the metabolic center of the neuron. Dendrites extend from the soma and are typically branched, forming a network that receives signals from other neurons. The complexity and branching patterns of dendrites are important aspects of neuron morphology. The axon is another critical component, extending from the neuron to transmit signals over distances to other cells. The morphology of axons, including their length, myelination, and branching, affects how signals are transmitted within the nervous system. And now we have the subcellular components. At a more granular level, neuron morphology also explores the subcellular structures such as dendritic spines, which are small protrusions on dendrites where synapses form. These spines play key roles in synaptic strength and plasticity. Synaptic morphology, including the size and shape of synaptic vesicles and the arrangement of neurotransmitters receptors, is also studied. This helps in understanding how neurons communicate and how synaptic changes can affect learning and memory. Neuron morphology is crucial because it influences how a neuron connects with other neurons, its role in neural circuits, and its ability to process and transmit information. The study of neuron morphology, which combines elements of anatomy, physiology, and biochemistry, provides insights into the functional capabilities of neurons and how they contribute to the broader functions of the nervous system. Neurons can be classified based on their structure into multipolar, bipolar, unipolar, and anaxonic types, each adapted to specific roles from processing complex information to transmitting signals over long distances. FEAGI is primarily designed around the multipolar type morphology and enables the creation of diverse neuronal projections and connectivity patterns. Neuron connectivity refers to the diverse patterns of connections formed between neurons, facilitating communication across the nervous system to support complex behaviors and cognitive functions. These include lateral connectivity involving horizontal links within the same brain area for local integration. Longitudinal connectivity for coordinating activities along the body or brain axis, feedward connectivity where information moves in one direction for hierarchical processing, feedback connectivity allowing for the modulation of input based on higher order information, convergent enabling the integration of information from multiple sources, divergent connectivity enabling the distribution of signals to multiple targets, reciprocal or bidirectional connectivity for complex interactions and adjustments, vertical connectivity, integrating information across different layers of structures like the cortex. Together, these connectivity types illustrate the brain's intricate wiring and its capacity for processing a vast array of information from sensory inputs to advanced cognitive tasks, showcasing the fundamental role of neuron morphology and the connectivity patterns in defining neural function and behavior. Neuron motifs are recurring small-scale patterns of interconnections between neurons within a network. These motifs are considered the building block of neural circuits and can be thought of as specific arrangements of a few neurons connected in ways that are statistically more common or functionally significant within the brain's network. Examples of motifs include chains, rings, and all-to-all connectivity among a small group of neurons. The study of motifs is part of the network theory and helps researchers understand the fundamental principles underlying neural circuit design, information processing, and the dynamic behavior in the brain. FEAGI, inspired by the diversity in neuron morphologies, has introduced a novel way of implementing diverse neuronal connectivity patterns and motifs using simple definitions of connectivity rules. Connectivity rules is a powerful tool that enables each neuron to assume a specific morphology that in turn enables it to transmit information to its neighboring neurons in a distinctive pattern. We will explain connectivity rules in detail later in the next tutorial.